Hello everyone, this will be lesson 2 dedicated to telling you all about the structure and function of proteins. This is the assignment brief, or adapted and taken from the assignment brief, so you know what we're going to be doing in this session. Basically you've got two jobs. You need to be able to outline the chemical structure of protein and then describe what it does in the cell membrane already. Now you should have a very good idea of what it does in the cell membrane already, because it was a feature of your cell biology test and it was a feature of your cell biology assignment, so you really have a good idea. But what we're looking for is a bit more technical detail about those things. So you might have, if you think about what the protein does in the cell membrane, you may well think, they're protein channels, they're protein receptors, they are the cytoskeleton of the cell membrane, they may well be enzymes, etc., etc. What I would expect you to be able to do after learning about this then is to look at the chemical structure of protein and link it to the functions that it does for the cell membrane. So this is what we're going to do: explain how amino acids join together. We're going to explain how they gain their specific shape, and then look at three functions of protein in the cell membrane. Now, this PowerPoint this video is really going to give you the bones for your assignment. It's going to take you through the key points of the assignment. You're going to have to go away and piece the different sources together and create the obviously the sentence structure and use diagrams, etc. But this will give you the fundamental knowledge and understanding to do well in your assignment. First of all, we've got to look at the chemical structure of an amino acid. Now, the good thing about these molecules is that they are chemically the same. So all amino acids have this amine group here, attached to a central carbon atom, and branching off it you've got a hydrogen atom, a carboxyl group, and the most special area of an amino acid is this R group here, which is sometimes called a variable side chain because it's only this thing that changes. Now R represents anything. It's just a letter which represents something that is there. Now it changes for each amino acid, and that's what defines what amino acid is. So every single amino acid that there is has this same structure here. It has a carbon atom in the middle, it has an amine group on the side, a hydrogen atom and a carboxyl group. But depending on which amino acid it is, it will have a different side chain or a different R group. They mean the same thing. And here, if you look to the right, this is a more chemical structure diagram, which shows you the bonds between the different atoms. Now, if you are familiar with my lessons on atomic structure and covalent bonding and ionic bonding, you'll see that there's a double bond there between the carbon and the oxygen, the hydroxyl group, etc. So it's fairly obvious. You can see that is the chemical structure of an amino acid. I would expect you to be able to describe that, to draw it, to make reference to it, because when we look at the amino acids of the human body, there are about 20. There are 20, and most of them that we use in the human body are taken in through the diet, and they're called the essential amino acids, and the others we can sort of synthesize them by the body. Now, what it's showing you really here is the chemical structure of each amino acid, and the only thing that is different is the R group or the variable side chain. So you can see if it was alanine, it would have this structure. If it was aspartic acid, it would have sort of this structure. If it was histidine, it would have this structure. But the only thing that you need to know is that the only thing that changes in an amino acid, in the different amino acids, is the R group. So you've got three nice examples here, hopefully, to spell it out for you. Here's cysteine. You can see that the core amino acid structure is the same as lysine, as it is as the same as aspartic acid. The carbon atom in the middle, the carboxyl group on the right, the amine group on the left, and the hydrogen atom at the bottom. Now you can see, hopefully, that the only thing that's changed is this R group. Okay, and you can see from the notes here that the R group can form disulfide bridges, which create cross linkages, which we'll come to in a minute. You can see that this side group gives it an alkaline property, this gives it acidic property. So you can see that the actual chemistry of it and the atomic makeup gives it properties of that amino acid. Now I'll leave it up to you to try and explain that if you so wish. It's not a vital, and I don't want to really confuse you. The major thing to get across would be that the R groups are differently, or structured differently anyway. So a protein is made of lots and lots and lots and lots of amino acids joined together, and this diagram really summarizes it nicely, because here we've got two amino acids. So we've got amino acid here, and we've got amino acid here. So in order to join two amino acids together, when we present them together, we have a chemical reaction, and this molecule here pops off. We've got a hydrogen from one of the amino acids and a hydroxyl from the other, and that pops off to make water, H2O. There's two hydrogens and one oxygen, and we know from our work on atomic structure, if you've done it, that is a happy molecule. Its electron shells are full, and therefore it likes to be together. But what we've done is chain these two amino acids together to make a dipeptide. A dipeptide, di meaning two. A polypeptide would be three or more. So basically we get the OH on that and the H on the next amino acid, join them together, and we've got a polypeptide, and so on. Now, if you're really good and you understand protein synthesis from our last topic in terms of translation, if you remember the ribosome, we've got these tRNA molecules bringing in the amino acids together on the ribosome, and then another one comes in and presents the next amino acid, and that one pops off, and we have a join there. And that's what we were drawing, in our kitchen activity in the classroom that we've been doing. And we said that basically another tRNA molecule, the ribosome, comes in with the corresponding anticodon, so that might be CCC, and we'd have GGG on the mRNA strand, and that would come along and bind to the next amino acid. Well, what's going on in terms of the binding of the amino acid? It's this reaction here, and we call that a condensation reaction. It's condensation because it is creating water, it's taking water out. It's sometimes also called a dehydration reaction. You might see that somewhere in the textbooks. Forgive my writing with a mouse. So this is called a dehydration or a condensation reaction, because the water from the hydrogen on one amino acid and the hydroxyl on the other amino acid is being taken out to join the two amino acids together, and that is how a protein is formed. That is what happens at tRNA during transcription. All clever stuff. So we call this simple chain of amino acids. Here, hopefully you can see it. It's an amino acid, amino acid, amino acid, amino acid. It's like a chain of beads almost. We call that its primary structure. And you can read this little box here from the handout, but all that's saying is that it's just a chain of amino acids joining together. And the beauty of this is, and I say it in class all the time, is that the primary structure of an amino acid determines the shape of the protein, and that's really key. If you, and we've seen that with sickle cell, if you decide to change one of these amino acids, it means that the shape, whereas it might shape up like this, it starts to shape like this, and you get a completely different shape. So the actual primary structure of them is really important, and that comes down to the transcription and translation at protein synthesis level. As the primary structure gets bigger, and we're starting to get hundreds of amino acids, they start folding up on each other, they start bonding with other atoms in the chain to form the protein's secondary structure. And it can form one of two shapes. It could be an alpha helix, so that little A sign with the two tails on is an alpha, or a beta pleated sheet. That's the two kind of shapes that amino acids fold into as the primary structure gets bigger and bigger. So here we've got the primary structure, and it's like, you know, we've seen the double helix before. It's like a telephone cord shape, which is what it says here. And hopefully you can see that here we've got the chemical structure of an amino acid. The chemical structure of an amino acid. And these are just amino acids that form this sort of telephone cord shape. It's just, you know, if we were to draw it in sort of ball term, it would be sort of like that for the amino acids, which actually, come to think of it, is drawn up there. But what this shows, which this top diagram doesn't show, that the bottom diagram does show, is these bonds here. Bonds here, and would be a bond there and there. They're the hydrogen bonds. Those bonds start to form between amino acids that aren't next door to each other in the chain. So if we look at this top picture, this is an amino acid chain all the way down. They're forming peptide bonds with each other. They're taking out water with each joint, and they're forming peptide bonds. But these yellow bonds here, these hydrogen bonds, which I've highlighted on the bottom diagram, are bonds between amino acids that aren't next door to each other. So I'm hoping you can see that yellow. It's not very good, but they're forming bonds between amino acids that aren't next door to each other in the chain. And that's what's making it three-dimensional. That's what's making it in the three-dimensional shape that you see right there. And if you put them in the same order every single time, you'll get exactly the same shape. That's why it's really important. And the same applies to the beta sheet. You get these amino acids that are chained together like this with peptide bonds, but you can see, hopefully, I'll do it in green this time so you can see it. These are the bonds that hold this beta sheet in its sheet-like shape. Try saying that after a few. And it's the hydrogen bonds that hold them together. I hope that made sense. It's mainly the secondary structure is about three-dimensional shape, and not the primary structure relates to chaining the amino acids directly together, whereas the secondary structure relates joining amino acids together that aren't next door to each other, that have that peptide bond. They form hydrogen bonds. So as the protein gets bigger, what we do see is that we get a mixture of beta sheets and alpha helixes. So hopefully you can see that here on this diagram here, you've got the alpha helix and the beta pleated sheet. It's just a complete mess of alpha helixes and beta sheets that form together to make, again, the three-dimensional shape. And it gets extremely complicated, but if you put the right amino acids together in the right order, you're going to get the same shape every time. And even if you put in 1,000,000 amino acids together, you put them in the right order, you get the same shape. So the tertiary structure of a protein is just a combination of alpha helixes and beta pleated sheets. And you will not be surprised to know, last but not least, the quaternary structure of a protein is again a mixture of tertiary structures. Now, like this text says, I think somewhere, some proteins stop at the tertiary structure, but a lot of proteins have a quaternary structure. So it's basically lots of tertiary structures together forming a humongous protein, and that is that. So when we're looking at the proteins on a cell membrane, you've highlighted these in yellow. There are so many, and your task is to sort of trying to explain the role of proteins with quality. And what you're really trying to do is to relate the structure of what we've just been through to the functions here on the membrane. Now you can see that some proteins are stuck to the surface of the membrane, some are channel proteins, some are receptor proteins, or they go all the way through. Some have carbohydrates attached to them. So there's lots of different functions of the protein on the cell membrane. You've even got the cytoskeleton, which prods in, and you've got tight junctions which seal cell membranes to cell membrane, like sealant. But the major aspect for you to get across in your assignment, and I'm going to let you choose a couple of these to do well, is how does their structure relate to function? And really we're talking about shape. Can you explain that their chemical structure relates to the shape and therefore relates to the function of what it does? What research can you do to find some examples? And that's the challenge for you today, is to try to go away and find some sources that support what we've said and this information is pretty much in any biology text, but you pick up at the right level how amino acids join together to form the primary structure, and how the primary, secondary, tertiary and quaternary structures link together in a specific way to do a job. Now hopefully this will be clear for you to follow. Feel free to ask me any questions.